So now as we continue to study photosynthesis, what we have to consider is where is all the ATP and NADPH that we're generating from the light reactions going? And it's going into the light-independent reactions, right? So again, we see the light reactions on the left, generating ATP and NADPH, and we're going to feed those into what's called the Calvin cycle, or the light-independent reactions. Sometimes you'll heard them referred to as the dark reactions. So again, to summarize, the light reactions are going to split water, release O2. We're going to reduce NADP to NADPH, and we're going to generate ATP from photophosphorylation. Now, light-independent reactions, in summary, then are going to form sugar from CO2 using the ATP and NADPH that we got from the light reactions. This is called carbon fixation, and it's going to be basically incorporating the inorganic CO2 from the atmosphere into organic sugars. Right? So it's expensive. It takes a lot of ATP and a lot of NADPH, and we're just going to look at a summary of the dark reactions in this course. Now, carbon enters the cycle as CO2 and leaves as glyceraldehyde 3-phosphate, G3P, which we've talked about before in glycolysis, and we'll come back and I'll show you that in a second. But basically, for a net synthesis of 1 G3P, you've got to turn the cycle 3 times, fixing a total of 3 CO2 molecules. It's not that simple or straightforward, but that's the math. So every time you turn the cycle, you bring in 1 more CO2, you get 1 more carbon, and you need to turn the cycle 3 times really to get a net increase of 1 G3P. So it has 3 phases: carbon fixation, which is going to involve the most abundant protein on the planet called ribisco. We'll talk about that. Then you have a reduction phase, and then you have to regenerate the CO2 acceptor, which is ribulose bisphosphate. So when we look at this, what we're going to see is 3 carbons, and we're going to see a 3 in front of everything, that prefix. But we're going to look at 1 carbon at a time, basically. So when in the presence of ribisco, what we're going to do is take that carbon and force it onto ribulose bisphosphate. Right, so ribulose bisphosphate is already a 5-carbon sugar, bisphosphate, 2 phosphates, and we're going to make it a 6-carbon sugar. That 6-carbon sugar then is going to break down in a very short amount of time into 2 3-phosphoglycerates. Right, so now I've gone from 3 6-carbon sugars to 6 3-carbon sugars. Right, so all the carbons add up. Now we go into the reduction phase, where we're going to move things around, phosphorylate, use our reducing power of NADPH to ultimately generate glyceraldehyde 3-phosphate. So 3GP, or G3P rather, is going to be the output. So from the 6 glyceraldehyde 3-phosphates that we can generate from this 3 carbons that have entered, we're going to remove 1 of them. Okay? And when we remove 1 of them, that leaves us with 5 G3Ps to play with. And in that process that is energy intensive using ATP, this regeneration step, we're going to regenerate ribulose bisphosphate that can be then ready to accept carbons again. All right? So this chart is really all I want you to know, and it is a vast, vast simplification. And if you look at the numbers, again, you can see 3 carbons coming in. We get 6 times 3 carbon molecules. We remove 1 3-carbon molecule. Now we have 5 times 3 carbon molecules, and we go to 3 5-carbon molecules. So it's a whole lot of rearranging of carbons and bonds. And again, that's relatively expensive. It takes energy. It takes reducing power. But basically, because the light reactions are driven by sunlight, which in essence is an infinite resource, it doesn't matter. It doesn't matter how expensive it is, because you can just collect more photons. Now, just to give you an idea of what we're not talking about when we look at this, the Calvin cycle in this summation, is this is just the regeneration phase of ribulose 1,5-bisphosphate. So it's a relatively complex process that we summarize into a single blue arrow that swoops around from the bottom of the circle. But if you take a plant physiology course, then you get into this level of detail, which, again, you do not need to know. But what I do want you to remember is that the fact that these biochemical pathways, they overlap a lot. All these intermediates can be on several different pathways, and even the pathways, major pathways like cellular respiration and photosynthesis, can overlap, right? So remember, our output ultimately is glyceraldehyde 3-phosphate, G3P, which we saw before in glycolysis, right? So remember glycolysis, we have our investment phase and then our output phase, and if you look, glyceraldehyde 3-phosphate is the very first molecule in our output phase. So if we feed this immediately into glycolysis, we're reaping rewards. We're reaping NADH and ATP immediately from the light-independent reactions of photosynthesis. You can also obviously build that up then into larger starch compounds and break them down later. But again, this is glycolysis. These are the same molecules that we also see in photosynthesis. So again, these biochemical pathways, the intermediates, are important to know, because then you start to understand how these things are linked and how life can modulate by shifting resources from one pathway to the other. So again, in summary, we have photosynthesis, these two major reactions, the light reactions and the light-independent reactions, ultimately making sugars, carbohydrates, as our end product.